Okay. Well, thank you, everybody. Welcome. And as I said, feel free to ask questions as we go. I want it to be clear to everybody. This first page that I want to show you is just kind of interesting stuff about what we're really talking about. The terms get used interchangeably. In fact, most people just say COVID whenever they're talking about something. But let's dig through this a little bit. The technical name for the virus that we're talking about is severe acute respiratory syndrome coronavirus 2, SARS-CoV-2. And I'm going to look for one other thing, get my laser pointer. So this is the name of the virus, and here's why it's named that. It's called corona because it looks a little bit like you've got jewels on the crown, and it's a virus, so we call it coronavirus. The SARS is due to the fact that it can cause severe acute respiratory syndrome. So we go COV, coronavirus, and we call it 2 because there was a coronavirus 1 a few years back. And then the COVID is the disease, the illness you get from that. And it sprang up notably in 2019. So again, people use those terms, do you have the COVID virus? They use them interchangeably. Actually, no one says SARS-CoV-2 very often. They just say COVID. But if you're interested, that's the name of the virus. And this is the illness that it causes. And I'll let you read that first line there from Khan Academy. From a biology point of view, and of course I teach biology here at Clark College, this is interesting because although we talk about killing it, a virus isn't alive any more than a stone is. So it's kind of funny to say killing it. It would be best perhaps to say to destroy it or damage it. But they are fascinating in that they're a little bit life-like, and what they're really good at is reproducing. And this is right here we have a cell, and this is a cell that's been hijacked by the coronavirus. And once the virus hijacked this cell, it sort of forced the cell to make a bunch of copies of the virus itself. And so each of these are viruses being sent out by the cell. Cells send out a lot of things. You know, cells in your pancreas will send out insulin. Some cells will send out hair. There are a lot of things that cells will do. And what the virus will do is it will tell the cell, I'm here now, I'm in charge. It takes control and it makes them make copies. So that's why what the cells are very good at is making copies of themselves. Wonderful image that's been colorized by Elizabeth Fisher. So here, let's dig into viruses just a little bit more. All three of these images here are viruses, and you see something on the inside. Usually it's coiled, and that will be either DNA or RNA. And when I say DNA, you all say, oh, that's the stuff that's in our cells. It is the stuff that's responsible for making us. That is correct. But what's interesting about these viruses is they have DNA or RNA and almost nothing else. So they're almost like just a carrier of this molecule, DNA or RNA, that is designed for reproducing. So in our chromosomes, human chromosomes, we have DNA. It's designed to make more cells, or another body if it's an egg cell or sperm cell. So, but these viruses aren't complete. They don't have the machinery. If you think back to high school biology, they don't have a nucleus. They don't have a ribosome. They don't have a bunch of these organelles. They just got a package of instructions. They'll have a protein shell, and inside they'll have this reproductive molecule, either RNA or DNA. Since they aren't complete, they aren't fully alive, they can't live on their own. They are obligated to parasitize something. They have, if they're going to reproduce, they have to take someone else's cell and say, would you make copies of this for me? It reminds me of me when I try and get something on my copy machine. I can't really do this. I just brought a thumb drive. Can I stick that in there and you do it for me? That's a little bit what the viruses do. So they come in, they put their thumb drive in your cells, and next thing you know, you're making all these copies that you really don't want to make. I like the image. Yes, please.
Sorry, I'm just wondering, if it is a non-living organism, how does it have the capability to replicate itself?
It cannot replicate itself without infecting a cell of a complete living thing.
Got it.
If you had virus, if I had some virus and I had a little paper and I knew they were on there and I didn't let them come in contact with any other cell, they would die. But if I managed to get them in me, then they're like, oh gosh, I can finally reproduce. Thank you, Mr. Clark.
Could I ask about the monoclonal antibodies and where that fits in with stopping this process?
Monoclonal antibodies. Oh, yes. I'm not going to get that technical, but we will come to that. And those are basically, that's part of the immune system that identifies these guys, say, hey, that's not part of your body, Steve Clark. If you don't mind, I'm going to go ahead and kill those. And I say, okay, great. My immune system will do that. That'll come later.
Okay, thank you.
So if you look on the right-hand side, viruses are not at all uncommon, but often we just go by a shortcut. Oh, that person had polio. That dog had rabies. But they're actually rabies virus, the polio virus, the HIV virus. And the reason, this is kind of interesting too, and something that I find really cool from a biology point of view. The reason this image over here is pointing to different areas are because these viruses, when they get in your body, they only get into certain cells that will that they can hook onto. So the dengue virus, it can best hook onto certain cells that are in your brain, but it won't hook onto cells that are in your lung like the SARS-CoV virus will. I'll talk about that more, but the virus needs to gain entry, and it sort of has to hijack a specific to itself cell, so that you'll end up with entry points that are in different parts of your body, and then symptoms that are going to be different. Each virus has a characteristic shape, no surprise there, and a distinctive shell. And there's a little factoid. Some of you know Rosalind Franklin. She was a big figure in science when they were trying to figure out the structure of the DNA double helix. And there have been some documentaries on her, and she was one of the early people who was able to look at very small things with enhanced microscope type looks. And we see this and say, oh, in the biology world, so that looks like a virus. She's the first one who was able to identify it. Just kind of fun to see her pop up in another area. Okay, so you know a little bit about virus. You know right now that they can't reproduce on their own, but you might not really know what that means. That might be kind of squishy. Let's look at what happens here. So this is a slide that I'm showing called how a virus would replicate. I put that red arrow in there because I want to remind you about something that isn't shown but is very significant. So here's what happens. We've got billions of cells in our body. And since we're talking about COVID, I'll go ahead and assume that this cell right here, this purplish one, is in my lung. So I inhale the virus. So here's the letter V in sign language for virus. I inhale it. It goes down into my lung, and then it attaches to that specific molecule or cell that we see right here. So here it comes. And what it does is it takes, and you can see in the middle of this one, it looks like that little twisty thing. That's supposed to tell us that it's DNA. RNA is almost similar. DNA has 2 strands, RNA has 1. So the virus gets in, and that's by foolery. The cell doesn't want it, but it sort of picks the lock and gets in here. And then it takes see how in this image they've covered that little section of DNA sort of reddish? And now they hook that red DNA right into the native DNA of the cell. Do you see that? If you are sick with COVID, for the weeks that you are sick, in your cells, in your lung and nose and some other places, you actually have a little strand of nucleic acid, of RNA in your body that does not belong to you. It's from the virus. So that's what they do. It hooks that little section of nucleic acid, DNA in this case, and then the cell, it doesn't know that it's foreign. So the cell uses the DNA and say, oh, I'm supposed to make a cell here. I'm supposed to make a whatever, some collagen. I'm supposed to make some insulin. I'm supposed to make, and it says, oh, looks like I'm supposed to make this little round thing, and it's a virus. And so this cell gets going, reading that, sending it out to a ribosome and making a whole copy. And then let me backtrack a second. You see this happening. This cell has been hijacked and it's making all kinds of viruses. And certainly at this point, the person is sick. Definitely they're sick by then. But that's what happens. Now this cell for a while probably does all of its other things as well. But some of these viruses have a way of really prioritizing their job, which is to make a new copy of them. And that causes the cell to shut down a little bit. Now you can imagine if your lung cells are shut down a little bit, or your nasal cavity cells are shut down a little bit, and people with COVID say, hey, I can't smell so well. But what a virus does is it comes into a cell, it hijacks it, and makes a bunch of copies. Now let's look at this red arrow. You guys look puzzled. Ask me a question.
I have a question.
Please.
So I hear people talk about the viral load a bit, and I was wondering if that, like, from what you just described, it sounds like one little piece of virus hitting one cell could potentially cause someone to be sick. But is that if you inhale more and more? I guess, what is the viral load and how does that relate to this?
Yeah, if I got one of these and it made all of these copies, even though I'm over 60, so I don't have the greatest immune system, I'm going to win because I only got one. But let's say you're a nurse, you're a doctor, and you're in the COVID area, which is every hospital, right? They are not getting one. They're getting so many that they better have great masks and great protection and a nice immune system because they're getting such a heavy load. It's a little bit like a poison. Poison is a function of dosage, right? If you take something that's poisonous, that taste it, ooh, you get a little bit, it won't kill you because you got such a small dose. That's the viral load.
Gotcha, thank you.
The initial load. And what my body will try and do, as soon as it identifies, it'll try and tamp it down. And if I'm successful, I don't get very sick. Think of children. But if I'm not very successful, I'll get very sick. And if I can't keep up with the virus, by that I mean my immune system can't, then I die.
I have a question.
Please.
Okay, I read this article in, I don't know, it was February. It was in the Wall Street Journal. And it was an article about two doctors they were from Turkey, but they went to work in a company, biotech in Germany. And one of the doctors on January 25th, he read a study that convinced him that the obscure disease in China would soon engulf the globe. He took 30 years of cancer research and ran 10 possible coronavirus vaccines. One ended up being the Pfizer. My question is, how do you take 30 years of cancer research to cure cancer? And is cancer a virus? I mean, what is it, the way cancer works in a cell that made this 30 years of research eligible for the Pfizer vaccine? I'm just curious.
It's a big question you're asking. And here's an answer that will fit our timeline.
Okay.
Number one, cancer isn't a virus. Cancer is a problem in your own DNA that causes your own cell, not a viral cell, to reproduce over and over and over and over, and then you grow a tumor. Sometimes a virus can cause that mistake to occur, but cancer itself isn't a virus. The research that people have done in so many biological fields it's amazing when you read about the vaccine, how many little nuggets from this research contributed, and nuggets from this research, and nuggets from this research. There were even researchers who were looking at lipids and fats, and that's part of the vaccine. So it is such a all hands on deck sort of phenomena that there are a lot of areas in biology that contributed. And that's about as well as I can treat that in this amount of time. But it doesn't surprise me what you said, that person would say, hey, this reminds me of some work I read here, or reminds me of cancer, or reminds me of anyone who's dealt with RNA. It all makes sense to me.
Thank you.
My pleasure. So now let's take a look at this red arrow, because there's one thing that this picture isn't showing, and that is, how does this virus fool the cell and get in the first place? Here's the answer. Let me show you my little study guide here. So if I were to say, oh, hey, here's something, we could view this as a cell, that's okay, but this is a lot better. Why? Because the surface of cells are not smooth. They have all kinds of things sticking out of them, all kinds of carbohydrates, proteins, a bunch of things. It's a busy place on the outside of the cell. And for a virus to fit in, they actually have to bind with one of the proteins that's poking out of your cell. So here's the surface of a cell. Something is poking out of it, and it has to have a certain shape that fits. And in human bodies, we have what's called this green molecule. It happens to be ACE2. There are a lot of them in our lungs. There are a lot of them in our nose. They're on the surface of our heart. They're in different places, not everywhere, but different places. The COVID virus happens to have a spike protein, that little pink part, and it fits like a hand in a glove. And as soon as it fits, the ACE says, oh, I know you. You must be a certain hormone that we've been expecting. Come on in. And the virus says, and in it goes. If you were born with a mutation, you didn't have that ACE2 receptor, you wouldn't get the virus. It wouldn't come in. In order for that cell to come in, it has to have something to hook onto. Our cell surfaces are a barrier. They keep things out. But this ACE2 receptor has a certain shape because it's looking for something valuable to come into that cell, a particular hormone. And instead, it gets something that's shaped similarly. It's the spike on the COVID. It gets fooled and says, come on in, my friend, we're waiting for you. We have ACE receptors in our lungs. Consequently, COVID is a lung impacting disease. We have ACE receptors in our nose. Consequently, it impacts our nose, but you don't die from a nose problem so much as you die from a lung problem. So even though it affects both places, it's more severe in the lung. But everyone who understands how you get sick by viruses has to understand there's got to be this little hand and glove that pulls the virus in. And for us, there it is, that little green thing. Here's a more complicated picture. I'm just going to walk you through it because it's the same thing. Here's the virus, SARS-CoV-2. Here are these ACE receptors, so we can think this might be a lung epithelial tissue. And we can either take the entire virus in, and then it will dump out its RNA and say, go ahead, start making a bunch of little viruses. Or sometimes the virus will just land and it won't come in entirely. It will just sort of open up and dump in its RNA. Either way, once the RNA, or with other viruses, DNA, once it gets in the cell, it says to the cell, would you please make these for me? Will you copy these for me? Here's my thumb drive. Make a bunch, make millions. And your cell does it. So this is the entry point. And this is actual COVID, or these are SARS-CoV-2 viruses that have been produced by both the blue and the purple cells, and they're coming out. Not dissimilar to what we saw before. But now you understand that first they went in, then they multiplied, replicated by the thousands. I don't know if that's true, maybe the hundreds per cell, but a lot of multiplication. Then they come out. And then they're going to hook onto another cell, or you're going to cough and they'll come out, or you're going to exhale and they'll come out. But these little viruses will float around. They're all over. So here's where we are now. COVID virus is in your body, and it brings with it that funky corona spike. That's the key. The spike is the thing that hooks onto the ACE2 receptor and gets drawn in. If your immune system could identify that spike, and it will, after being sick, why does the person recover? Not a vaccinated person, just anybody. Why do they eventually recover? Because as soon as you get infected, your body has these immune system cells, and they start bumping into this COVID thing. They say, hey, I had this spike, that's funky. I don't think that's part of you. I don't think that's Steve Clark. I think that's something different. Hey guys, come over. And they kind of put a mark on it and they say, let's kill it. And that's where the question about the antibodies, the monoclonal antibodies, they'll come in. So once the spike is in our body's immune system starts to look for it. It starts to recognize it. And then the cells from your own body that are fighting this disease, they multiply as they should. Like the first day I get sick, I've got a few immune cells. The second day I'm sick, I've got even more immune cells, and they beef up to try and take care of things. And at 2 weeks, they can take care of things. At 2 weeks, you'll have enough of your own fighters inside your body that you can defeat that thing and survive. However, of course, 2 weeks is a long time. And in that time, if you've got an underlying condition, if you've got a weak immune system, if you're older, you might not make it, or you might get grievously ill. So that's what happens naturally. What naturally happens is you've got this spiky thing roll around your body, and your own immune systems, T cells, they'll be looking around and say, hey, we don't recognize that as Steve Clark. We've been in his body for 60 years. We haven't seen that. I think we should kill it. Here's what happens. This is the normal immune response. Harmful things enter your body. The Y's are antibodies. That's what my body produces. All our bodies have them. They're floating around all the time. And though these things, they look like they're all the same shape, but they're not. They actually have millions of different shapes. And what they do is they say, hey, the surface of this bacteria looks funky. I want to mark it. I think we should kill it. Hey, the surface of this thing, this virus, looks funky. It doesn't look like Steve Clark cells. I think we should mark it. And as soon as it's marked, other cells come in and they kill it, and you get well. But it takes 2 weeks. This is what happens normally. It takes about 2 weeks for these antibodies in your own body, your immune system, to get so powerful that whatever came in, they'll crush it. Now, if you have a low load, like Katie suggested, maybe they'll crush it sooner. But on the outside, it could take 2 weeks for your numbers to really ramp up. How are we doing? You have a question? Well done, my friends. Once the invader is marked by your antibodies, other immune cells come and kill it. When you are done being sick, when you're feeling healthy, you might say to yourself, well, I guess all these antibodies and all these immune cells, they can just, die off like an old blood cell. Yeah, they do. But some of them, what we call our memory cells, our memory immune cells, some of them sit in your lymph nodes and they sit in your spleen and they wait. And that way, if you ever get this same virus or the same bacteria a second time, they are ready and they just jump. And it doesn't take 2 weeks, it takes 2 or 3 days. That's why we have these memory cells. That's why when you were vaccinated for polio when you were a kid, if you get, it would be not unlikely that your body has already killed the polio virus 10 times in the last 20 years.
Wow.
Because you've got those guys waiting. And as soon as they see them, as soon as they sniff them, boom, they ramp up quickly. Look
I
how
have a
quickly.
question.
Yeah,
So it's public disclosure. It doesn't make any difference. I got my vaccine. And my response to the Pfizer vaccine was really swollen lymph nodes. And in my neck, especially, and under my arm. So I called my doctor and I told her, and she said, that's good. She says, you've got a really healthy immune system. So my
Yes.
question is, with a vaccine, because I had such a strong response to the vaccine, will, if I got it again, even though I'm vaccinated, now I know I can get it again because it's happening out there, right? People are
Yes.
vaccinated and
yes,
they're
yes.
passing, yeah. So will I have the antibodies the same as if I didn't have the vaccine? Will my antibodies be less strong because I didn't do it, just on my own? You know what I mean?
I do know what you mean. I do know what you mean. I'm not going to answer that because that's part of this lecture coming up. So I'll do it when
Okay.
it comes up. But yes, I know exactly what you mean. Thank you.
Okay.
Here's what happens the first time you get something. It could be a bacteria, it could be a virus, who knows what it is. The first time you get it, here's my 2 weeks. You have an immune response, and by that means your cells actually increase. You get more antibodies, you get more of these. They actually are called natural killer cells in your body. They find every spike that's floating around anywhere in your nose, in your heart, your colon, your lungs, all of them. They kill them. And then after about 20 days, you start feeling better. And some of those guys go and they hide in your spleen and they hide in your lymph nodes. And then let's suppose a year later you get it. Those guys in the lymph nodes and the spleen, they come out. Look how big the response is. And look how fast it is. They get to this level in 5 days or 4 days. So the second time you get something, you're like, I think I got something. 2 days later, I guess I didn't. Oh, you had it, but your body just crushed it. That's what the immune response is. What the vaccine wants to do is fake your body into thinking you have it so that you get this response, so that you don't have to have this 20 days. And the more dangerous the virus is, the more dangerous these 20 days are. You can die in those 20 days. But if you're vaccinated, or if you had it before, then the chance of you getting gravely ill is reduced because you've got this gigantic response. That's what your body does. So that's what we want. So vaccines try to fool your body into making protective antibodies without you having to get sick. That you could do it either way. But wouldn't it be nice to get the same sort of protection, this protection, without having to have, be intubated in a hospital?
Right.
Stephen,
Yes.
sorry, based on what you just said, are you saying that for those who have had the virus in the past, that the vaccine would be redundant because their body would already be set up to have the same response?
Yes and no. The short answer is yes. There are 2 ways to get safe from the virus. Get COVID, because it's just like the measles. You're not going to get it twice, right? So you get it yourself, or you get a vaccine. The problem with that is that studies in the last year have started to show that having both is even better. And that if there's another thing, and that is that these are studies that are suggesting, it's not 100%. And this kind of gets to the question you were asking before, Lori. If you had a low load, you had a small infection, your body stamped it out, there's a chance that your body didn't get quite as well prepared for this as it could have. So your fundamental question, Carrie, is yes, there is some redundancy to that. But with a real serious thing, you want to make sure that you're all set to have a fast and powerful response. That's why they're considering boosters for some people, because there are some weaknesses in both the virus and the natural infection. Both of them have a little bit of weaknesses. Okay, so
I have a question.
May I also, do we have different variants as well? So that maybe if you got something that was not a Delta variant earlier, you don't have that antibodies for Delta variant that's out there right now. So it's good to have the vaccination anyway.
yeah, and it would also, it's interesting that it wouldn't surprise me. I'm just thinking off the top of my head, this isn't science, this is just me. It wouldn't be surprising if people who are making vaccines are saying, I wonder if we should make a Delta variant vaccine. It's almost the same. You still have a spike, but the spike on the Delta variant is, take a look at my hand, it's got a little funky shape on the spike. And the Delta variant with that funky shape, it fits even better to the ACE2. So yes, you're right on that. I am going to speed ahead for a couple things because the one concern is that this be shared with other people and they might not be as curious as you, and I don't want them to get slower than they need to. So the idea with vaccines is to fool your body into thinking that you've been sick, and therefore it'll make these protections. It'll give you the memory cells. It'll have you all ready without being exposed. And there are three different approaches to making vaccines. One is you use a whole virus. I'll explain all these. Another one is you use parts of a virus. And this most recent one is, hey, what if we take not all of the RNA or DNA, just the part responsible for making the spike? So if that's Steve Clark, we would take the DNA that's responsible for making the shape of my ear, not all the rest of the stuff, just the ear shape. Because that's what your DNA or RNA does. It has a little section that's the recipe for this, there's the recipe for that. So they would take a little chunk of it. So let's look at what these three things are. This is a traditional vaccine. All of us who have gotten our shots in school, we've had this kind of vaccine. What they do is they take a full virus. Doesn't that make you scared? Because they're going to inject that. But then they do something they'll like take X-ray radiation, and you see this nice, beautiful RNA, DNA, it's damaged. They put this in your arm, and you've still got these guys that are lock and key. Your body will recognize these, and they'll kill it. But since they've destroyed the RNA, DNA, that virus can't do anything to you. So that's one, what they call whole, but inactivated. The RNA, DNA is wrecked. That would work because it still gives your body a chance to recognize these things and make antibodies. That's one kind of virus. It's difficult to make it when you identify it, but it can be done. Here's another one. I'll let you read that first sentence. In this, they take a virus and they say, we believe it's this chunk here that is really gaining entry into your cell. So we're going to break off these chunks. We're going to put those in the vaccine, shoot them in your arm, and your body will see those chunks and it will respond to that and you'll have immunity. Here's another one. This is a little bit funky, but for the non-sciency, they'll take a live pathogen like a virus, dangerous. They'll put it in a non-human host, a lab rat. They'll leave it in that population of rats for a year. And with the virus in the rats for a year, they'll get variants and they'll evolve. And after a while, they'll evolve so that their shape doesn't fit quite so well to humans. It's less pathogenic because it's evolved away from what it fits the rat great, but not the humans anymore. Then they'll take that weak pathogen, and they'll put that in your arm. That's another strategy for fooling your body. But this last one, that's what is fascinating from a science point of view. They're going to take, again, not the whole strand. If they took the whole strand, we wouldn't be, you'd just be getting COVID. They just take a chunk of it, just the part that has the recipe for the spike. And it's faster and it's easier. You can scale it up. You put that little chunk of DNA or RNA into a vaccine, that wrap it in a lipid. So they don't actually just insert RNA into your arm. There's RNA, and it's sort of like, kind of like giving your dog a pill in a pill pocket. They'll swallow it more easily, and it makes it so that your body doesn't immediately attack the RNA because it's wrapped in this little bubble of lipid. Then that little chunk of RNA will go into a lot of your cells. I imagine the same ones that have the ACE receptor, and it will make just the spike. It won't make a whole virus. It just makes a spike. But even so, when that spike shows up in your body, your immune cells say, hey, spike, we don't like spike. We're going to kill you now, and we're going to remember you for later. Bingo, you have it. And you never have the virus. All you have is a funky little spike. I got the vaccine in January and February. I got spikes. Glad to have them. I don't have any of the other RNA, just a spike. And my body's memorized it. So that's what I love about this. I mean, the other viruses, they've worked. Polio is in decline, but it feels nice to know that it's not attenuated. It's not a whole thing. It's just a little chunk of spike. I'm in good shape. And then, as I said, your body stores those memory B cells that will recognize any spike like that, and I'm safe. Here's how the vaccine looks. I think you can understand this now that we've gone this far. So here's the virus. They take just a little piece of that RNA. And by the way, they can manufacture that RNA in a lab in a petri dish now. And then they'll put it in a little lipid fatty bubble that is in the syringe. That goes into your arm. It finds its way to your places where you have the ACE receptors, nasal cavity, lungs, the pericardium around your heart. And then those will end up with little spikes on there, and that will activate your body. Hey, wait a second, what is this? They identify it, these antibodies, they make more antibodies, many, many more. They kill all those cells that had a little spike, which is why you felt a little bit sick after you got the vaccine. Your body's going to work. We're going to kill this. You could say, hey, it's not really a virus. You don't have to kill it. We're killing it. Okay, well, I don't feel good for a day. But they're not even killing any virus. So yeah, a doctor says, hey, you feel a little swollen? You feel a little uncomfortable? Oh, great. That means this part is working. Your body's responding. Yeah. And then the key thing is they hide out in your body. And then the next time you get that big reaction. I almost said spike. That probably wouldn't work. You get this big reaction and your body makes you well. That's the nuts and bolts of my talk. I'm going to run down a couple pages of things that are kind of quick and easy. Why are vaccines safe? Now, if you guys are showing this to your class in a couple weeks, you can just stop at this point and look at them. But this is sort of what they do first, second, third, fourth. You have, so I'm not going to talk about it carefully, but first they'll study how they can maybe make a vaccine work in an animal. Then they'll ask for human volunteers. Then they'll do, I see I got to do a little spell checking here, small group human trials. They call those clinical trials. Then they do the large ones, 30 to 60,000. And then this I love, this is so nice science. Then the results of those. So Pfizer has these groups of 30 to 60,000 people taking the vaccine, and they don't let the Pfizer execs look at it. They have a third party of scientists look at the results. They don't have any investment. And they say, what do you think? And if they see something troublesome, like everybody who's left-handed seems to get real sick from this, then they stop it. So they have a third-party look, and then there's a different committee that makes a recommendation to FDA. Then initially they have this emergency approval, and now it's a huge trial because it's millions of people, right? And they're still looking, and then they get the final approval. So I view that as robust, and it makes me as a science guy pretty comfortable.
Even a couple slides ago, you had the RNA, DNA viruses, and maybe it meant to say vaccines are new. Can you define what new means? Is that just at the beginning of when COVID started, these are new? Because my understanding is we've been studying RNA vaccines for a long time, and that's why we were able to adapt and create the vaccine quickly. So define new for these purposes, please.
So yes, this slide should have said RNA, DNA vaccines are new. So good catch, Laura. The viruses are as old as the hills, but the vaccines are new. And by that, I mean, when we got vaccinated in the past, we were vaccinated by this kind of vaccine or this kind of vaccine. But to actually take a little chunk of the RNA or DNA and make that be the core delivery into the body, that is new. The first time that that's been done on a large scale is now.
Stephen, do you have time for a question?
Let me go through my last slides and then I do.
Okay.
This is interesting and this is kind of new. There are more ACE2 molecules. They're in higher density in our nose than in our lung, which is why if you've got a mask over your mouth, you could be exhaling all those viruses that are replicating in your nasal cavity. They knew that the ACE2s were in your nasal cavity, but they didn't know they were at a higher density. I don't actually know if that's recent news, but at any rate, that's a big deal. So when you have your mask on, it makes it so the amount of virus that escapes naturally just floating on your breath is significantly lower, and they're subject to gravity. So they'll fall down to the ground and not be inhaled so easily, which is why you have the 3 feet, 6 feet social distancing. Singing and yelling can propel droplets farther, of course. You know, we've all had these uncomfortable dinners where you look across at your father-in-law and in that particular light, you say, oh my goodness, there's a lot of fluid coming out when he just tells a joke or laughs or talks. Oh, well, on the other hand, that's what life is, and that's what's made us healthy for millennia. But you can see these things travel, and masks are helpful for that reason. So I think Clark is really following the good recommendation there. I started to call this factoids, but it's not good to call science facts because we want to get the idea that everything is revised when you have a better study. So the Delta variant is, the spike has a slightly different shape, like I said before, and it fits more tightly. And it also has a little feature in the Delta variant that makes it replicate a little more quickly than the originals. The virus that's floating in the air can be there for 3 hours. On a solid surface, it could be there for 3 days. But on a solid surface, that's not going to be a problem unless I somehow coax those onto my into my mouth or nose. So it doesn't seem to last long on clothing. And the one hypothesis is that the heat will damage some of the aspects of the virus. This is related to the question that I think Katie asked. You can have, if you get an infection with a low dose, you might be asymptomatic, but it still can help you be immune. And this is the answer to a question I think Lori asked early on. The vaccines seem to, recent studies, show that their secondary infection response is more powerful than if you got it naturally. And children haven't been studied much. If for some reason the world said this is a children's disease only, they would have focused everything on the children and they would have had this vaccine ready for children. And then they would have said, we're not approving adults yet because we haven't studied them yet. So it's really just a function of who has been studied first, who was prioritized. And children seem to be more robust. And also, there's this idea that, you know, if you're going to do something experimental, let's do it on people who can consent, people who are older. So often they'll start with adult volunteers, and it's not surprising that children are last to get these things, but they are in the same process, just not out of the tube yet for children. Thank you. I wish you a spleen full of memory cells. And you can take off whenever you want. And this would be a time when people at Clark, if they feel like, hey, I'm done with it now, that would be fine. But otherwise, feel free to ask me a question.
Thank you, Stephen. But the question that I had was, you indicated that RNA and DNA virus vaccines are new. And one of the, one of the fake news things that people have brought up against this vaccine is that it affects your DNA and it alters you genetically. That's one of the biggest ones I've heard out there. Well, we don't know what this is going to do to us in 2 years or so. And meanwhile, we're getting all these variants that could be even more dangerous as the virus mutates. So is there any concern that these vaccines, I mean, from what you said, I don't feel that, but I mean.
No, let me jump right in. No, the people who study every time you get sick with a virus, your DNA has changed. That's how it works.
Right.
Every time you get sick. So that happens routinely. And maybe if you didn't know, you would be nervous about it. But for people who know these things and understand, who study these things, they're not greatly concerned by that, nor would I be. Don't forget also that when you take the virus and the cell that makes that spike, that little cell with the funky DNA on the inside now, it gets killed.
Yeah.
So your body is always, your immune system is always busy looking for something in your body that isn't right. And that could be a surface protein, or it could be some DNA on the inside. And your immune system is constantly looking to change those. But every time you get sick with a virus, you have some DNA change. That's part of the human condition. That's not an unusual or no, it doesn't bother me.
Okay.
Steve, could I ask a question?
Yeah, Terry.
Thanks. I just read about the dogs in the airports that can smell the virus, which it's a two-part question, because the real concern I've got that I keep reading about is that it's going to continue to mutate, and in underdeveloped countries where they haven't been able to get the virus or the vaccine. I mean, I don't want to be pessimistic, but how is this ever going to, it's just going to continue to mutate unless we do something like with the dogs and stop people.
Yeah, your first concern is a good one. And so let's imagine we had a little petri dish and you had a colony of bacteria growing and you did something to it that allowed them to mutate. Of course they would. The mutation rate would be pretty slow. They live in one petri dish. But if they're infecting 1 billion people, think of how many petri dishes that is. Yes, whenever you have a rampant pandemic, I guess that's redundant. Whenever you have a pandemic, evolution is happening at a much higher rate than if it were an isolated illness. Absolutely. So what would have happened with a pandemic 500 years ago? Well, a lot of people get sick, and the only way out of it is the infected people die, and that reduces the number of petri dishes, or the immune systems in the people they have a broad enough response that they can kill several of the variants at the same time, which also isn't unlikely, but it is costly because people die, you know, as that response is being coaxed out of all the humans. Yes. And I don't know how the dogs smell it. I don't know how they do. I don't know if they smell the virus, which I actually doubt, or if they smell some of the symptomatic responses of humans. I don't know. I think it is known. I just don't happen to know that answer. It's fascinating.
Thank you.
Okay, my friends, I'll call
Can I
it a
have
wrap.
one last question?
All
One
right.
last question. You said a few moments ago that the virus probably doesn't live on our clothing because of the heat or the temperature.
That's
That's one of,
only a hypothesis.
okay,
The scientists noticed that the virus doesn't live very long on clothing. And then they asked, that was just an observation. And they say, why? Why would it not live on clothing? You know, what is it about clothing? And the hypothesis was whenever you have heat, molecules move faster. And when they move faster, they're likely to get a little bit of damage. So that was a hypothesis, but I don't think it's known why they don't survive on clothing.
okay. I was just wondering if the cold weather would, because cold weather, you'd seem to get the flu more but that's because people are inside. So I didn't know.
There are a handful of different reasons why cold weather tends to be a higher flu time. Yes.
Okay.
And some of those are social reasons like human behavior.
This has been great. Really. Thank you, Stephen.
My pleasure.
Thank
Bye,
you.
everybody. Thank you.
Thank you.
Thank you.